This conference
before,
will now be recorded.
and the first class I just given about, I've told about what we will learn during the entire course, and the second class we have learned about what is molecular biology, what is the scope of molecular biology, brief history of molecular biology, then what are the constituents of DNA, what is nucleoside, what is nucleotide, and nitrogenous base, how nucleoside attaches with nitrogenous base, and then how the polynucleotide forms, right? So I think so. We will go for revision. Okay, we will go for quick revision. Whatever we have learned. So we have learned that what is molecular biology. Molecular biology is that it simply is the study of interaction between the DNA, RNA, and protein at the molecular level, and the DNA is also known. We came to know that DNA is a deoxyribonucleic acid, and it is the hereditary material which passes from one generation to other generation, um, and it's nearly same the constituents of the DNA or the composition of the DNA varies from person to person, but each and every cell in a person body has the same DNA and same type of DNA in a single person. But if we compare with other person, there will be 1% differences in the DNA sequence. Okay. Now, what is nucleic acid? We know DNA and RNA is called as nucleic acid. Who have isolated the first who have coined the term and first isolated the nucleic acid, that is DNA. The scientist was Friedrich Mister. Both of these, this DNA and RNA, both of these are also known as polynucleotide. Okay, now how this nucleic acid, this DNA and RNA is made up of? Okay, so we have seen that it is made up of DNA is made up of your DNA RNA is made up of nucleotide. The nucleotide consists of three things: nitrogen base, sugar moiety, and phosphate bond. Phosphate, right? So now nitrogen base is attached with a sugar moiety with a glycosidic bond, and then this the five carbon atom of the pentose ring, the ribose ring, is attached with a phosphate group with a phosphodiester bond, and thus it forms a nucleotide. And these nucleotides are attached with another nucleotide by a phosphodiester linkage and forms a polynucleotide. And so the polynucleotide is what? Is the arrangement of repeated manner of repeated number of nucleotides, right? And it forms a polynucleotide. Okay. Now the polynucleotide, each polynucleotide strand forms a double helix structure of DNA. How it forms? It forms with the help of a hydrogen bond formed between uh, which the bond which is formed between the bases. The that bond is called as the hydrogen bond, which is formed between the bases. And so we came to know that A bonds with T and G bonds with C. Till that we doesn't know that whether these bonds, whether A bonds with T and G bonds with C is compulsory or is a natural or is a random process. So but in 1950, one of the major inventor of that time was the Erwin Chargaff, who have postulated the uh, Chargaff's rule. Okay, and which become the basis of the elucidation of the DNA structure, which is elucidated by Rosalind Franklin, Maurice Wilkin, and James Watson, Francis Crick. Okay, we'll go through one by one in this class. So I hope you people are doing well, keeping yourself safe, and utilizing this time. So one of the best, I like this quotation which is given by Marie Curie. Okay, so remember this for for getting in for getting involved in science is science is not the subject for mugger. Science is the subject, is like you have to make like a hobby, then you will feel like science is very interesting. Okay, so nothing in life, Marie Curie stated that nothing in life is to be feared, it is only to be understood, right? We don't have to be feared, but if we can understand, the fear will go. Okay, now whatever we are doing, now is the time to understand more, so that we may fear less. So it's completely based on our lifestyle. Okay, and now we are in young age, so few people will be in BSc, well, some will be in MSc, someone will be preparing for competitive exam, some will be, uh, a few students will be preparing for PhD entrances, competitive exam. So we all have fear that how to qualify, right? So this is the time you can understand more and learn more and reduce your fear. So be safe, utilize this lockdown period completely, and study hard to reach your goal. Okay, so now today in today's classes, we will learn about that, what is the Chargaff's rule, how Chargaff's rule help in elucidating the DNA structure. We will, today we will not going to solve some mathematical problem regarding the Chargaff's rule, which usually comes in competitive exam. In the next class, I will be I will be solving some of the important numerical questions of, which usually comes in competitive exam like CSR, JNU, ICMR, uh, based on Chargaff's rule. So in the next class, I will be solving few questions. So this class, we are not going to do that. We will go through a theoretical class today. Okay. So now, then we will learn how DNA structure came into light, what the brief history about the uh, elucidation of the structure uh, coming came from the light. Okay, so came to us finally in 3D structure, or three-dimensional structure, 2D two-dimensional structure. Okay, whatever what you are seeing, how it came finally. Okay, because the structure of DNA was very controversial in the period of 1950, 1940 to 1950. But after 1950, the DNA structure came, which is means made by James Watson and Francis Crick. Okay. Now we will go through silent features of Watson and Crick DNA model, which is normally known as B form of DNA, and then we will learn about other important forms of DNA, which usually exist in the cell, but very, you can say very few seconds, or very short period of time, and that type of DNA is aDNA and Z form of DNA. Okay. So now Chargaff's rule. Okay, Chargaff's rule. He is the another important contributor whose finding provides a valuable foundation for Watson and Crick DNA model. The Erwin Chargaff, in 1950, he published a paper in which he states that adenine, number of moles of adenine is equal to the number of moles of thymine, and similarly the number of moles of guanine is equal to the number of moles of cytosine. And, and he also stated that the ratios of adenine to thymine and uh, guanine to cytosine are always remain close to 1. It never crosses 1, and nearly this ratio is same in all species studied, and this finding is now known as Chargaff's rule. Okay, you can see the picture. He is the when he was working in the King's University. This picture was taken on that time. Now, after his publication, now three important principles, or he postulated three important principles. That is, the amount of adenine residue is proportional to the amount of thymine, and same for guanine to cytosine. Okay, which means that A is equal to T and G is equal to C. Okay. Now, second point he states that the sum of the purines, okay, sum of the purine A plus G is equal to the pyrimidine C plus T. Okay. Therefore, that is A plus G means the concentration of A plus concentration of G is equal to concentration of C plus concentration of T. Third principle is that percentage of C plus G does not necessarily equal to percentage of A plus T. Okay, uh, it tells that the concentration of C plus G in percentage, it it actually it does not always equal to the percentage of A plus T. You will see that the percentage of A plus T and percentage of C plus G always varies. Okay, and it depends on species to species also. The ratio, the fourth point is states that the ratio of A plus G by T plus C is equal to 1 for all species whose genome is double stranded DNA. Remember that this Chargaff's rule is only applicable for double stranded DNA and double stranded hybrid DNA or a double stranded RNA, but it's not applicable for single stranded DNA or single stranded RNA. Okay, now now from this all the 5 points, mathematically we tell that Chargaff rule can be expressed as the composition A plus T means adenine plus thymine plus guanine plus cytosine is equals to 1, where we came from the principles state that where A is equals to T and G is equals to C. Okay. Now the same you can see in the figure. um, A equals to T, G equals to the at time no one knew why why it is. I have told you that this Chargaff's rule on the Chargaff Erwin Chargaff only stated that A equals to T and G equals to T. Before that, no one knew why it is happening. Okay. Now we know it's because adenine is always hydrogen bonded with thymine, and guanine always bond across with cytosine. And we came to by reading from that principle, that 5 principle which I have stated, that the percentage of A is equals to percentage of T and percentage of G is equals to percent of C. In it will be always equal in when you compare in the double stranded DNA. Now, how this double stranded DNA, that is Watson and Crick, how he elucidated this structure and how he make the model. Okay, so First, the first scientist, actually, if you'll go in deep, then the most important person was the Linus Pauling, who was working at the California Institute of Technology. Okay, he he elucidated that he elucidated the chemical bonding for the alpha helix structure, and this alpha helix structure is stabilized by hydrogen bond. Okay, and this information led the formation of double helix model, that is Watson and Crick. This was the one group which was working on that time. Another group was that Maurice Wilkins, Rosalind Franklin, and their colleagues were trying to find out the structure of DNA using a X-ray diffraction picture, which is taken by the Rosalind Franklin in 1952. Usually, if we'll read about the history of that how Watson and Crick built the ball and socket model, DNA model, uh, the three-dimensional model, you will find that Watson and Crick never performed an experiment by themselves. Their tactic was to use other groups' data to build a DNA model. They have never done the experiment. They have just taken the information from this group, that the by which is by the X-ray diffraction pattern data from this Franklin group. And this Franklin X-ray work strongly suggested that DNA is formed of two chains. Okay, you can see in the figure in the right side that Rosalind Franklin is there, and in the right of that, and the pattern you can see dotted dots are there. It's like X form. Okay, this X form is of the because of the two strand, and it's forming a helical like structure. Okay, you can clearly tell, okay, no, the DNA is like it is made up of two chains and wound in a helical pattern, and it is the simplest form. So this insight, uh, this X-ray diffraction pattern data also reveals some important features of the DNA structure, that it also tells about the size and shape of the helix. Okay, shape of the helix as shown in the figure. Okay, they have elucidated that the shape of the that it is of 3.32 angstrom, the repeat distance is of 3.32 angstrom, and the whole helix ton is 33.2 angstrom. And this information finally shared by the Maurice Wilkins, one of the scientists who works with Rosalind Franklin. In 1953, he shared the diffraction data and the picture with James Watson in London. Okay, and in 1953, after getting the information, Watson and Crick finally built or resolved the double helical structure of the DNA, which is commonly known as B form of DNA. So you can see that James Watson is standing and pointing out something in the DNA model, and Francis Crick was observing that. Okay, so it has an interesting story behind it. And then they got the Nobel Prize. This Maurice Wilkins, James Watson, and Francis Crick shared the Nobel Prize, but actually this Rosalind Franklin died on that time, so she did not get the Nobel Prize, because the Nobel Prize is not given to the dead person. Okay, so now they outline this all the information from the Rosalind Franklin X-ray diffraction data, and then they outlined this model features in the journal Nature Journal in 1953 and stated the following features. And they stated that the two long polynucleotide strands are coiled around a central axis in a right-handed helical fashion. Okay, the two polynucleotide chain are coiled around the central axis in a right-handed helical fashion. The second feature is that these strands, these two strands interact with each other. How these strands interact with each other? These two strands interact with each other by hydrogen bonds formed between bases. That is, G forms 3 hydrogen bond with C and A forms 2 hydrogen bond with T. The third point, the helical structure is uniform. Okay, this helical structure is uniform, having sugar phosphate backbone on the outside. Okay, it has sugar phosphate backbone on the outside, and the bases are inside, and these bases which are in inside interact these two strands by forming a hydrogen bond between them. Fourth point is they states that the two strands are in anti-parallel nature, which means that one strand runs in the 5' to 3' direction, and the other strand run in the 3' to 5' direction. Okay, so now fifth point, they states that angle of interaction between base pair is there is some angle, there is some interaction between the bases also. Okay, and there is some angle which generates because of this arrangement of this nitrogen base or nucleotide um, in some particular angle, it creates one major groove and minor grooves in the DNA. Okay, now sixth point, the spacing between base pair. Okay, the spacing between base pair, it is also called as helix rise per base pair, is 3.32 angstrom. Means the two base is separated from each other in the distance of 3.32 angstrom. The overall helix repeat distance means it is also called as helical turn along the axis or helix pitch is about 33.2 angstrom, which means that one helix turn consists of a length of 33.2 angstrom. Okay, it is 33.2 angstrom long, one helix turn. Therefore, from the above two information, you can easily you can calculate that per turn of the helix consists of 10.4 base pair. Means 10.4 base pair means 20 nucleotides in both strands. Okay. So you can see in the figure. The figure A shows the anti-parallel nature of the two strand. See, one strand is running from in the left side, the arrows goes down. So 5' it runs from 5' to 3'. The other end runs from 3' to 5', and the arrows goes up. So it is an anti-parallel nature, and it's you can see the anti-parallel you can see the anti-parallel nature forms a little zigzag. Okay, zigzag hydrogen bond, and this gives because of the bonding between the nucleotide bases in some angle. Okay, now in the figure B you can see the diameter of the helix in the down, it's 20 angstrom. Okay, this is the B form of DNA. The diameter of the B form of DNA, which is postulated by Watson and Crick, is 20 angstrom. Now you can see the minor groove, okay, major groove in the picture, and you can see this is called helix turn. One helix turn consists of 33.2 angstrom length. Okay, now the spaces between the bases is of 3.32 angstrom. uh, Now this is a field diagram of the structure of the DNA. Okay, you can see the my major group and the minor group, how it forms. Uh, so now to make it more clear, uh, I will show you that this is the major group formation and the minor group formation. There is tilt. This is called base pair tilt. Is minus 6 degree. This base pair tilt, this base pair tilt, this is the angle which generates major group and the minor group. Okay, and this base pair tilt is usually because of the formation of the hydrogen bond, and this hydrogen bond is not formed complete perpendicular to the central axis, but it is tilted at minus 6 degree. And this base pair tilt, you will find that in A form it is different, in Z form it is different. Okay, now this helix pitch, which I call, which is helix um, per ton of the helix, is called as helix pitch, which is 33.2 angstrom. Okay, now 10.4 in one helix pitch, yeah, one helix ton, it is having 10.4 base pair per ton. Okay, now the different forms of DNA. Okay, what are the different forms of DNA? This whatever we have learned till now, it is of your B form of DNA. Okay, which is explained by Watson and Crick, based on the Chargaff's rule and Rosalind Franklin data. But more in the cell, this type of DNA is you can find mostly, but in some cases, but for short period of time, there is two forms of DNA which usually exist. Okay, so that is a form of DNA and that form of DNA. Okay, so now a form of DNA. Okay, how this a form of DNA forms and where you can find, and how it is different from B form of DNA. So a form of DNA, you can see in the table, uh, that you can see the repeating unit, helix sense, rotation per base pair. Um, the clear differentiation is there. Now, how it exists? The A form of DNA exists when we reduce the relative humidity surrounding the sodium salt of DNA fiber to 75%. Okay, means when the relative humidity of the surrounding, see, the X-ray diffraction pattern, which is generated by Rosalind Franklin, was a sodium salt of DNA in crystal form of DNA. Okay, and it was surrounded by a humidity of 92%. So that B form of DNA exists in that type of condition. But scientists found that if we reduce this relative humidity surrounding this sodium salt of DNA fiber to 75%, the A form of DNA exists. Okay. However, in a cell, such forms usually seen in hybrid polynucleotide. Hybrid polynucleotide means what? It contains one DNA and one RNA in a solution. What that means? In a cell, two polynucleotide will not form a form of DNA. Usually, it is found, the structure is found usually in hybrid condition. It's one DNA and one RNA with base pair tilted away about 20 degrees centigrade. 20 degree. Okay, from the central axis. Yeah. Now, as compared to the B form of DNA. Okay, you can see in the figure that base pair tilt, A form of DNA, A form of DNA having 20-degree base pair tilt, means 20-degree tilt means away from the central axis. Now, but in B form of DNA, it is having minus 6 degree. Okay, now the not only that, okay, there is also a several differences which you can see, that is in case of diameter, our DNA, A form of DNA is wider, that is having a it is wider than the B form of DNA in diameter. So you can see in diameter, it is A form of DNA is 23 angstrom. Okay, while B form of DNA is 20 angstrom. Correct. Now, this helix, helix per pitch, this helix packs in 10.7 base pair. Means the base pair per turn consists of 10.7 in case of A form of DNA, but in B form of DNA consists of only 10.4. Okay, so it having a more number of base pair in a turn. Okay, because of this, the pitch or the distance required to complete one turn of the helix is 24.6 angstrom. Okay, where in B form of DNA it is having 33.2 angstrom. Okay, similarly you can see the helix rise per base pair. Okay, along the axis is 2.3 angstrom. Helix rise per base pair along axis is 2.3 angstrom in case of A form of DNA, while in case of B form of DNA, 3.32 angstrom. Okay, now the glycosidic bond is the glycosidic bond is similar in A form of DNA and B form of DNA, but the sugar buckling is different in A form of DNA and B form of DNA. In A form of DNA, the sugar buckling is happening in C3 indo, in carbon 3 position, while B form of DNA shows sugar buckling at C2 carbon 2 position in indo condition. Okay, then we can see the differences in major groove and minor groove. The major groove is narrow and deep, and your minor groove is wide and shallow, while in B form of DNA, major groove is wide and deep, and minor groove is narrow and deep. You can see in the figure in the left side, you can see clearly that the major and minor groove differences in A form of DNA and B form of DNA. And there is some similarity between the A form of DNA and B form of, both are right-handed helical structure. Okay, so this is about A form of DNA. Now jet form of DNA. Okay, similarly the jet form of DNA is also different from the B form of DNA and even from A form of DNA. Okay, now the jet form of DNA is mostly found in left-handed helical structure. Okay, because of alternating purine pyrimidine sequences. Okay, it has a repeated nature. The repeating unit is of it consists of purine and pyrimidine only, and because of this repetition, okay, this left-handed helical structure exists. Okay, and it forms a hydrogen bond in completely zigzag manner. The backbone is not uniform like B form of DNA. The backbone follows a zigzag path, okay, as compared to B form of DNA and A form of DNA. Okay, then further Z DNA is thinner. Thinner means the diameter of the Z form of DNA is thinner. It is of 18 angstrom only, while as compared to the B form of DNA and A form of DNA, the B form of DNA having 20 angstrom and A form of DNA 23 angstrom diameter. So Z form is thinner than this B form and A form of DNA in case of diameter. Further, the repeating unit I told you, the repeating unit in A form and B form DNA, in both the form of DNA, is one base pair. Okay, because it has 4 different base pair arranged. But in that form of DNA, the base pair are arranged in dinucleotide. That is also in the nature is alternating purine and pyrimidine nature. Okay. Further, the sugar and glycosidic bond conformation is completely different from A form and B form of DNA. Okay, that is it has a specific for purine and a specific for glycosidic conformation. It's specific for purine and specific for pyrimidine. So you can see is the glycosidic bond is anti. Anti and syn means if then the hexose structure in the nitrogen base is in the right side. Okay, it is called anti. If it is in the left side, it is called syn. Okay, so this anti is the glycosidic bond in jet form of DNA having a anti conformation for purine, and for pyrimidine it has a syn conformation. Okay, now you can see there is also differences in the sugar coupling. Okay, this purine ACT having a C2 indo. Okay, while G and C is while G is having a C3 indo. So in this case also they have as the conformation is different for purine and pyrimidine. Okay, now the major groove and minor groove. Okay, because of the zigzag nature, major groove mostly flattened. You can't see, usually you can't see in the figure. You can see in the left side jet form of DNA. Okay, it's completely flattened, but you can somehow you can see the narrow groove. Okay, the minor groove is narrow and deep. It's completely narrow and deep. Okay, so these are the differences in the formation of uh, A form of DNA and Z form of DNA by in comparison to the B form of DNA. Okay. Now there are other form of DNA which I will not explain here. Maybe I will explain in my YouTube channel. That is triple helix DNA structure. Okay, then quadruplex DNA structure. Okay, then I will explain about propeller twist, base tilt. Okay, all this I will explain in my channel. Okay, not here till now. So these are the things for today. The next class, the next class, we will learn about we will solve some numericals uh, for Chargaff's rule. Okay, and then we will proceed for our further information, further classes, which will be taken.
Okay, sir. Okay, sir. And now I would like to ask students if they have any questions, please come and ask. So, Srivalli is asking, sir, what is sugar pakading, sir?
Okay, so wait. Yeah, what was the question?
What is sugar pakading?
Hello.
Yes, sir. What is sugar
Okay.
pakading, sir?
See, yeah, yeah. See, sugar puckering means see these sugars. Usually, we think that this sugar for moiety, which is a ribose, in case of DNA and RNA, because this is other ribose having a ribose sugar. Okay, we think that it is a planar in nature. Okay, these are not planar in nature. Okay, the these are the 5 carbon atom. Okay, the car see uh, usually what happens, this 5 carbon atom means pentose sugar. The oxygen position representing the top of the, if you will think like an envelope. Okay, if you think like an envelope, so when you open the envelope, it form you can see it will form like a pentose sugar, okay, ribose sugar, it will form the envelope. Okay, so in that form you can see that it is not a planar. Okay, the oxygen can be bent out of the plane of the envelope. You can bend that uh, what to say, envelope, that opening part you can bend. You can bend to the right side or you can bend to the left side. So this twisting happens in carbon 2 and carbon 3, okay, relative to other atoms. It is, which results various twist forms of the sugar ring. Okay, now when if we'll see from the above, if carbon four carbons, okay, 1 2 4 5 will be in the same plane, and carbon 3 will be towards you. Okay, it will be towards you, it means that it is an upside, then it is called as carbon 3 endo. Okay, similarly, if the carbon 2 of the pentose sugar is towards you, it means that C2 endo. This uh, this rotation of the ribose sugar twist, twisting of this of the carbon atom in this ribose sugar usually happens in carbon 3 and carbon 2. Okay, this twist is only known as sugar coupling. Am I clear? or
Okay, sir.
or I will or I will explain in next class if you people need it.
Okay, sir. Okay. And the next question is, why the ratio of adenine plus guanine to thymine plus cytosine is equal to 1 only for double-stranded DNA? Why it is not constant in RNA?
Yeah, it's a good question. Okay. See, usually this, if we will calculate the percent of adenine and guanine and A plus G and T plus C is in case of RNA, you will find that it will not be equal distribution. Okay, because it does not have a complementary pairing. Okay, it does not depend upon the complementary base pairing rule. Okay, the complementary base pairing rule is what? A will bond to the T and C will bond to the G, and vice versa. Okay, but in the case of RNA, you will not find this complementary base pairing. So that's why you will not get the the composition of A plus G plus T plus C equal to 1. Okay.
Okay, sir. Okay, sir. And the next question.
It's clear. I think Sunidhi asked this question, right?
Yes, sir. Uh, any more questions, students? If you want to ask, you can ask now. Please come and ask. What is relative